Now, it's very important to have a small idea about, or a brief idea about the cell cycle. You know that the cell go into different phases of proliferation. Originally, we have the quiescent cells, those in G0, they are sleeping cells, ready to do something. And when the cell go into proliferation, it goes into different phases. And these phases, from G0, it will go into G1. In the G1 phase, it is the phase of preparation of proteins and RNAs in order that the cell will go into another phases in the process of growth and division. And we have the S phase. The S phase is the phase of synthesis of DNA. And we have G2, or pre-mitotic phase. It is again continuation of the protein and RNA synthesis, and we have the mitosis or the cell division. Why we need this? Because some of the chemotherapeutic agents working only in specific phases. We say for these chemotherapies that they are phase specific. Some of them, they are not phase specific, but they work on cells who are proliferating in the cycle regardless of the phase. For example, alkylating agents work on the different phases, okay? And we have chemotherapeutic agents, rarely, probably occur, work on G0 and other phases. So that it's very important to know which kind of chemotherapy. Sometimes in the principles of using chemotherapy, we give it multiple drugs. One of them, they work independently on the cell cycle. One of them will work on this cell in the cycle regardless of the phase, and one of them will target a specific phase of the cycle. One of the important principles is the tumor angiogenesis. The tumor unable to survive unless there is blood supply for them. Okay? And those are capable, the malignant cells are capable to induce their own blood supply. So that the tumors, in order to survive, okay, they need a network of blood supply. If the tumor in 1 or 2 millimeter, probably it will be provided by nutrition from nearby tissue. For example, ischemic graft. Ischemic graft in the first 48 hours, it takes nutrition from the recipient bed, okay, by imbibing of these nutrients to the cells. And after 48 hours, a blood supply started to develop in this area, which make the cells making the ischemic graft to survive. So that if a tumor going anywhere and it's proliferate and unable to produce good nutritional support, it means that it's going to die and to fade away. So that the lack of nutrition, lack of glucose, lack of oxygen will induce apoptosis of these cells and their death. If we take this slide, we find that those are cells affected. There are multiple heads exposed to carcinogen, and they start to have a well-established malignancy in that area. They start to secrete angiogenesis factors in order to make network of vessels supplying them. Okay. Once they have these vessels, the blood will go to the tumor and it will start to survive. Usually, those blood vessels are not of good quality. They are thin walled and they can easily penetrate by malignant cells. So that the malignant cells, they invade their own blood supply. So that we say, for example, that there is a vascular invasion. When we have a breast cancer, we read the report, they said to us that there is a vascular and lymphatic invasion. Vascular invasion, it means that the presence of tumor tissue within the lumen of blood vessels in the same tumor. And this, once it reaches the blood, it may migrate through the network, okay, anywhere in the body. As you see, that those will go into the blood vessels, and those malignant cells, when they go in the blood vessels, some of the blood elements, like the platelets, the fibroblasts, will wrap the tumor cells, collectively making what's called malignant empress. And this will go from one vessel to another, and it is growing in its size and protected from immune mechanisms to be targeted until reach their destination, final destination, where they cannot move more, so that they will be embolized and fixed to the terminal vessel, as you see in the third picture. And then once they are there, they start to extravasate and to migrate from the blood vessel to the neighboring tissue. Okay. Actually, when they go there, probably they will acted upon by immune mechanism and it will be killed. So that there are certain areas where they can survive. There's a good soil in that area, okay? Namely, the areas where we all the time look for metastasis in them. For example, the bone, okay? For example, the liver, for example, the lungs, for example, the brain. So that those are good sites for malignant cells to establish new life in new place. Why we, and so that you see that after going in their final destination, they start again making new blood vessels and they will repeat the story. One of the major advances, okay, like when we know this mechanism, the people start to work on trying to cut the blood supply to this area. People may embolize the tumor so that it gets necrosed. People may give chemotherapy, okay, to reach these blood vessels based on the fact that those blood vessels are not of good quality, they don't contract. One of the techniques, for example, in the management of liver tumors is to cannulate the hepatic artery. The cannulation of the hepatic artery, the blood will go through this, the drug will go through the hepatic artery to the liver. Now, if we can use a vasoconstricting material, okay, namely use the glucagon, the blood vessels of the normal liver tissue will start to contract, but the blood vessels of the malignant tissue, because they are lacking muscular layer, they don't contract, so that the blood containing the drug will be directed to pool where? In the area of the tumor, because the blood vessels, they are primitive ones. This is one thing we benefit from knowing the angiogenesis. The major thing that they have anti-angiogenesis factor. Okay, there is a drug now called Avastin. It is used for management of colon cancer and breast cancer. It has an established benefit improving the survival when we use Avastin for management of these malignancies. And the main target is the blood vessels of the tumor. And as you see, these are the effect of the new vascularization of the tumor, perfusion of the nutrients, the newly formed endothelial cells stimulate the growth of the HS tumor cells by secreting polypeptides such as insulin-like and so on. And angiogenesis is needed for metastasis to occur. It is necessary biological correlate of malignancy. We will stop here.